Hi everyone, today we're going to talk neurobiology of disruptive behavior, developmental perspective, and relevant findings. So our goals for today, we are going to understand current perspectives on emotional behavioral regulation, identify the technological advances that link disruptive behavior and structures of the brain, generalize studies in animals to emotional regulation and behavioral regulation in humans, explain the role of oxytocin and cortisol in development of disruptive behaviors, understand the role of the HPA system in relation to cortisol and disruptive behaviors, explain the attenuation hypothesis, describe the prenatal influences on disruptive behaviors, and explain the sex differences in the development of disruptive behaviors. So ERBR is what they call emotional regulation and behavioral regulation. They don't really state it that well in the chapter, and it's just abbreviated. So let's talk predispositions. Predispositions are underlying genetic and neurodevelopmental predispositions to misinterpret emotional and behavioral clues that influence constraints on regulatory function. So basically what this is saying is that children sometimes have these predispositions that make it really difficult for them to control their behavior and their emotions. They're not able to develop those skills to actually constrain their behavior. Neurobiological predispositions could include temperament, and that could predispose to disruptive behavior. So if you think back to your basic psychology class and the different types of temperament. So we have kids who have an easy temperament, we have kids who have a difficult temperament, and we have kids who are slow to warm. So they're talking probably here about either the difficult or the slow to warm kids. Environmental. So early childhood experiences could either override or moderate genetic influences or genetic predispositions. So a child's environment could either make an expression of a gene worse, or they could lessen that expression of a gene. Another environmental predisposition could be modeling behavior. So if you have a parent who is either aggressive or they have problems with emotional regulation or behavioral regulation, the child could model their behavior after that and learn that from their parents. Epigenetics, remember, is the way that a gene gets expressed based off of influence from the environment. So a gene's function or expression can be changed without affecting the DNA structure of the gene. And in terms of epigenetics, context is really important. The context activates or dictates the expression of a gene. Genes are bound to that environment. And there's some family characteristics that we know are linked to the development of conduct disorder and oppositional defiant disorder, specifically harsh or inconsistent punishment. And there are some other factors as well. Perinatal complications, so complications right after birth, neurocognitive deficits, low IQ, and parental antisocial behavior all could have a predisposition for the development of difficulties with emotional and behavioral regulation. There have been many technological advances for disruptive behavior disorder, and the first is testosterone. We know for a while that testosterone has been linked to aggression. However, molecular structure, receptors, and the location of receptors in the brain have now been specified to add to the information that we have about that link to aggressive behavior. And they test this by assay serum or saliva concentrations, and they look not only at testosterone, but they also look at cortisol, and cortisol is our stress hormone. Structural MRI and maltreatment. So they've been able to tell that there's a difference in brain structures of those who are typically developing and those who are physically abused. They also can tell that there's differences in the brain to neuroresponses to anger. So if a child is in an MRI, usually a functioning MRI, and they're shown some type of anger stimuli cue, so either some type of picture of somebody who looks angry, or it could be an angry tone of voice or a raised tone of voice, they show anger differently than children who are typically developing. Our orbital prefrontal cortex is one of the most important aspects of humans. It is our executive functioning center, so it makes us who we are. It's responsible for our planning, our decision making, but it's highly vulnerable to environmental disruptions because it doesn't fully develop until we're 25. Maltreated children had smaller prefrontal cortexes, and the size of their prefrontal cortex could actually predict the severity of their maltreatment. The cerebellum is also implicated. It's influenced by the environment. It's involved in cognitive and social behavior, and it's also associated with our executive functioning. The posterior superior lobe is smaller in children who are post-institutionalized, so somebody who has either spent an extended amount of time at a hospital for maybe a psychiatric ward, or they might have been in some type of residential treatment. Their posterior superior lobes are smaller than typically developing children. Those children with those smaller posterior and superior lobes showed poor executive control, and they showed poor social regulation. So they had difficulty navigating a social situation. There are also electrophysiological measures. Children who are abused can identify anger more quickly because they're sensitized to this signal, because they devote a disproportionate amount of time and cognitive resources to these signals. So they're primed to these signals because they've been so frequently exposed to them. They attend to them much longer and much more severe than people who have not had these environmental factors. ERP and EEG, an event-related potential, is a type of measure on an EEG, and it's time-locked to a specific stimuli. One aspect of this, P3B, is a good measure of selective attention on a task. P3B showed that physically abused children had enhanced attentional allocation toward vocal expression of anger. So if they heard an angry tone of voice or a raised tone of voice on this EEG, they showed enhanced attention to this. They also showed enhanced response to angry faces. The P3B amplitude was in response to the severity of physical abuse. So if they were more physically abused, then they could see the difference on the amplitudes of this wave, because remember, an EEG studies your brain waves. Children who are maltreated also had difficulty disengaging or turning away from these angry faces. They had impaired regulation of goal-directed attention. So if they were given a task and they had these angry signals, either somebody who had that angry look on their face, or they had some type of auditory cue like a raised voice or an angry tone of voice, they were not able to complete those goal-directed tasks. N2, which is an ERP component, show that children who are maltreated will attend to angry faces even if they're instructed to ignore them. Task-related irrelevant angry faces, so having some type of angry face in the background that had nothing to do with the task that was at hand, predicted poor performance. The severity of maltreatment predicted the attention to anger. So children who were more severely maltreated tended to spend more time attending to these angry faces or these anger cues. The more time they devoted to these anger cues, the worse their performance was on a task. So if you think about that, that makes sense. If you're paying so much attention to these anger cues, you can't pay attention to the task at hand. And the more you're maltreated, the more likely you're going to be attuned to that, because we all know, unfortunately, if you're maltreated and those anger signals come up, you know that you're going to suffer the consequences. So it makes it harder to pay attention to your task at hand. Coherence of non-human and human models. So here we're going to talk about animal studies. Maternal behavior can affect change in response to stress in offspring, and it can change gene expression. So maternal behavior can either make the offspring, their babies, better able to cope with stress or less able to cope with stress. 5-HTT is a neurotransmitter transporter that fine-tunes transmission of serotonin by reuptaking it from synaptic clefts. So let's have a quick review of neurons. So you have a neuron that fires, you have the neural signal that goes all the way down through the neuron where it gets to the terminal buttons. Those are those pictures here. And because electrical impulses can't jump that synaptic cleft or that space, that gap, it turns into neurotransmitters. So the terminal button releases neurotransmitters, and then those neurotransmitters are picked up by the dendrites on the receiving neuron. So each type of neurotransmitter has a specific receptor. So the neurotransmitter is released, and whatever is not picked up by that specific receptor gets reuptaked. So it just gets reabsorbed back into the fluid that is in the brain. And remember that serotonin plays a large role in depression. So there's two types of alleles. There's a long allele, and the long allele has higher serotonin reuptake efficiency, so that serotonin isn't hanging out in the synaptic cleft nearly as long. In stressful situations, those with two long alleles can cope better with stress. The short allele has lower serotonin reuptake efficiency, and either one or two short alleles shows more fearful reactions to stressors. Monkeys who are raised in stressful situations with the short allele have impaired serotonin transmission, and in humans, the short allele is related to psychopathology. Brain development and emotional regulation. So let's talk about the neuroscience approach. The neuroscience approach focuses a lot on voluntary or effortful regulation versus automatic regulation. And when we're talking automatic regulation, there's neural circuits that either lessen the forms of negative affect once elicited, or they maintain positive affect. So these are systems that we don't have control of that either help us cope better by lessening the negative effects or maintaining our positive affect. And one of the most basic forms of an automatic emotional regulation is extinction. And extinction occurs when the conditioned stimulus is presented without the unconditioned stimulus, so that conditioned response stops occurring. So think back to Pavlov. So Pavlov trained his dogs to salivate at a tone, so he paired tone, then he gave food, and they salivated. Tone, food, they salivated repeatedly until he was able to just present the tone and the dog salivated in response. Well, what happens in extinction is if he presents the tone over and over and over again without the food, eventually the dogs are going to stop salivating because they're no longer going to associate that tone with the food. So this is what they're talking about in extinction. In this case, they're talking about sweating and other automatic responses that occur with stress. So rapid heartbeat, our breathing picks up. The extinction occurs when the pathway from the prefrontal cortex, so our executive functioning center, to the amygdala is blocked, so the amygdala response is lessened. And remember that our amygdala is in our emotional regulation center. It regulates all of our emotions, especially fear and aggression. So if that pathway is blocked, then those expressions don't come about as intensely. So let's talk brain structures. Abnormal amygdala and prefrontal cortex are associated with disruptive behaviors. So any abnormalities in those structures can predict some disruptive behaviors. The orbitofrontal, ventromedial, prefrontal, and cingulate cortex are all important in decision making, behavioral control, and emotional regulation. So these all have those areas that help us to regulate both our behavior and our emotions. Deficits in these regions may cause impulsivity and impaired moral judgment. So if that part of the brain is damaged, it is really difficult for children to make good decisions. The amygdala and the hippocampus are critical for emotional processing. Impairment leads to shallow affect and lack of empathy. And hippocampus and amygdala damage is found in studies with children with callous unemotional traits. We're going to talk about callous unemotional traits in the next chapter. There's a whole chapter that's devoted to that. The endocrine system also plays a part. When they're talking about the endocrine system, they're talking specifically about the hypothalamic pituitary gonadal axis and the HPA axis, the hypothalamic pituitary adrenal gland axis. So just to review, our HPA axis is our slower but longer lasting response to stress. The hypothalamus alerts the pituitary gland activates the adrenal cortex, and the adrenal cortex then releases cortisol, which is our stress hormone. Cortisol increases our metabolism and blood sugar. It allows us to be prepared for that fight, flight, or freeze response. Cortisol signals the brain to shut down the response once the stressor is removed. The endocrine system also deals with posterior hypothalamic hormones. One of these hormones is oxytocin, and oxytocin plays a large part in the reward circuitry. Oxytocin is also associated with bonding behavior, so it's associated with maternal bonding. It's associated with fidelity between people. If your dog stares in your eyes, they're releasing oxytocin. They're trying to strengthen that bond between you and them. I always tell my husband that my dog loves me so much, and he just wants some oxytocin released so he can stare into my eyes. Oxytocin, sorry, I'm going to pause while the dog drinks. Sorry, as I was saying, so oxytocin is associated with decreases in cortisol, and it is also associated with positive social interactions and, as I said, attachment behaviors. Oxytocin can lessen the effects of isolation or negative emotions and behaviors. Oxytocin increases empathy and empathetic tendencies, and it lowers our reactivity to stress. Post-institutionalized children have dysregulation in their oxytocin systems. They show a muted or a lessened oxytocin response after interacting with their mothers. So they don't have that reward circuitry or that bonding that typically people do have with their mothers. Voles are highly social and monogamous animals, and studies with voles show that oxytocin protects against behavioral and cardiac dysfunction in relation to chronic stress and cortisol. So let's review real quick what happens when you have chronic stress. So as I said, when your HPA system activates that cortisol, eventually your brain tells your HPA system to stop releasing cortisol once that stressor is removed. But if you have that chronic stress, then that cortisol keeps getting released. But that energy, that cortisol, is never burned because you never have a fight, flight, or response, fight, fright, or freeze response. So what happens is you have all that cortisol that still is roaming all around in the body, and what it ends up doing is it ends up depositing in the heart. And when it ends up depositing in the heart, it eventually builds up and it leads to thickened arteries and blocked arteries. And that's why stress is often related to cardiovascular disease. So oxytocin can help moderate that effect in voles. Cortisol does have an effect on energy, cardiovascular function, like I had just said, immune activity. If you're chronically stressed, your immune system is less efficient, growth, emotion, and cognition. Cortisol adaptively is released to cope with stress, but they're talking stress in the short run, not in the long term. Chronic cortisol impairs behavioral adaptation and is associated with difficulties in emotional regulation, behavioral regulation, and psychopathology. Those children who are post-institutionalized have dysregulation in their HPA system. They do have prolonged cortisol elevation after interactions with their mother, so showing that interactions with their mother is actually a chronic stressor for them. High cortisol levels are also associated in girls who have been sexually abused and children who have been severely neglected. Cortisol is going to be one of the most important hormones that we talk about. So let's talk specifically cortisol. So the HPA system, as we know, deals with our stress level, but it also has a role in development of disruptive behaviors. So what happens is that HPA response can get altered by inadequate parenting, early toxic prenatal experiences, abusive and insensitive caretaking, inconsistency of caretaking. It alters the development of threat, fear, and stress-relevant systems. So if we have that chronic exposure to those, we're going to be less sensitive to those type of events. And as we know, those type of events can lead to disruptive behavior disorders. So the fact that they have always been exposed to these events and they're less likely to respond to it means that they might develop some thrill-seeking behaviors, or they might have to kind of up their behavior in order to have that same input. The basal cortisol level is the amount of cortisol that is detected in saliva in the morning. And the context in which children are raised affects our basal cortisol level. Parental insensitivity predicted lower basal cortisol levels in teenagers. And we're going to talk about this in the next slide, but there is a contradictory or a paradoxical effect where basal cortisol levels, so cortisol is our stress hormone, so if you're chronically stressed, you would think that it was high, but in children with disruptive behavior disorders or parental insensitivity, all of those predispositions that could cause disruptive behavior disorders, their cortisol levels are actually lower than typically developing children. So the most important part of this chapter is the attenuation hypothesis. Basal cortisol level and disruptive behaviors. There is a negative association. Children with disruptive behaviors actually have lower basal cortisol levels. So like I said in the previous slide, it's a paradoxical effect. You would think that children who have chronic stress would have higher levels of cortisol since cortisol is our stress hormone, but it's actually the opposite. Children with low levels of basal cortisol are predisposed to externalizing and aggressive behaviors, and those with high levels of basal cortisol are predisposed to fear. And the attenuation hypothesis is the tendency for disruptive, externalizing, and criminal behavior to exhibit lower concentrations of cortisol. So those who exhibit those behaviors have lower levels of cortisol. Lower levels are the result of earlier experiences such as chaotic or inconsistent parenting, and it results in the downregulation of the HPA axis activity. So our HPA system, which is already slow acting already, acts slower. Individuals adapt over time when they're confronted with stressful situations, and our chronic stress decreases that stress response or the release of cortisol. So if our HPA system isn't working as well, then the amount of cortisol that gets released is going to be impaired. Allostasis is our response to stressors to maintain homeostasis. Chronic cortisol does not shut down after the stress is removed. So chronic stress, that cortisol response always keeps going, as I had said in a couple of slides ago. So that chronic cortisol actually damages our HPA system, and when it damages our HPA axis, we're not able to send those signals to shut down the cortisol level. Some other factors: minor traumatic experiences, insensitive parenting, more time in child care centers, and interparental conflict all are related to lower basal levels of cortisol. Attenuated cortisol, it's predicted that child externalizing symptoms over a 2-year period has been associated with attenuated or lower cortisol. It's associated with low empathy, callousness, sensation seeking. So sensation seeking will often increase the probability of disruptive tendencies and criminal behavior. They have low autonomic nervous system response, so that fight, flight, or freeze response, theirs is not as fine-tuned as most people's. It's associated with later criminal behavior. Sensation seeking at age 3 is linked to aggression. So again, because they don't have that cortisol release, they don't have that fight, flight, or freeze response. So they're not in tune with their levels of cortisol, and they seek other ways of getting that input, such as sensation seeking. Our HPA and our autonomic nervous system are affected by genes. They're also affected by rearing conditions, parental and family interactions. Those children with internalizing symptoms have blunted or lowered cortisol levels. Without typical HPA functioning that prepares people to handle these stressful situations, children develop poor coping skills that are associated with disruptive behaviors. Or it could be that blunted cortisol may be a pattern in families where conflict and violence occurs. So if you're constantly exposed to that stress, your HPA system becomes damaged and that cortisol never gets shut off, You just get used to it, so the lower level of cortisol is what occurs. When we're talking about attenuation in puberty, the timing of puberty and gonadal steroid hormone changes are related to individual differences in emotional regulation and behavioral regulation. The timing is important. Higher cortisol in later puberty was related to antisocial behavior and rule breaking. Lower cortisol in earlier puberty was related to conduct disorder symptoms, but these were only for boys. This does not actually apply to girls. Attenuation and sexual maltreatment. Basal cortisol levels increased steadily from middle childhood into early adulthood for those who had sexual maltreatment. The raise was less steep than non-abused girls, and this shows that girls who are sexually abused have neuroendocrine alterations that are characterized by low activity. So very similar to those who have disruptive behaviors, those who have sexual maltreatment also have lower basal levels of cortisol. So let's talk about some of the prenatal influences on neurobiological development. Teratogens, so that's anything that can cause death or harm to the baby. Teratogens show damage in both human and animals, and there's a couple of specific ones that we're going to talk about. Cocaine is one of them. Prenatal exposure shows structural and functional changes. The frontal lobe changes suggest a less mature brain. Cocaine exposure also has effect on emotions, obesity, neurocognitive memory, executive function, attention, and inhibition. So all of that goes along pretty much with your prefrontal cortex, the memory, the executive function, the attention, the inhibition. So cocaine damages that. The obesity part I'm not sure about. I didn't know that there was a link to prenatal cocaine exposure and obesity. Nicotine also is a huge teratogen that causes a lot of problems. It has negative effects on attention, decision-making, memory. It's linked to ADHD and motor, sensory, and cognitive deficits. So again, it's going to damage our frontal lobe, which is going to make us more prone to problems with executive functioning. Some environmental risks: maternal depression, hostile parenting practices. Young age at childbirth is also associated with the development of disruptive behavior disorders. Antisocial behaviors of the parents, the level of education, smoking, low family income, family dysfunction, and lack of stimulation all are predisposition and risk factors for disruptive behavior disorders. Stress during pregnancy also is one that affects disruptive behavior disorders. So stress negatively affects maternal and fetal HPA and HPG, the axis. Stress hormones are related to emotional regulation and behavioral regulation. And they have what's called low corticotropin releasing hormones in early pregnancy. It's related to depression symptoms, and it predicted later depression. Low CRH is related to a greater number of conduct disorders, and CRH levels are inversely related to depression. Low CRH indirectly affects fetal brain development. So in this section of the book, I actually found it really difficult to understand. At some points when they were talking about CRH and the effects, it was difficult to follow if they were talking about the effects on the pregnant mom, or if they were talking about the effects on the fetus, or if they were talking about the effects on both, and who exactly these affect in the development of the disruptive behavior disorders. So I thought that section was really quite unclear, unfortunately. I will say that that's probably the one thing that I dislike the most about this textbook, is it kind of assumes that you have this really strong background in genetics and molecular biology and molecular genetics. And I know, I've been teaching for 20 years, so it's been a while since I've been in college, but we didn't learn that type of stuff when I was going to school. And maybe the curriculum changed, or maybe it's a little bit different, because I went to school in Massachusetts. Massachusetts is not categorical like Michigan is. I just got a general special education degree, and I could teach, and I was certified in all disabilities. So maybe it's a little bit different, but I thought that last section was difficult, just like I found all of chapter 2 difficult. So let's talk steroid hormone fetal milieu and temperament. Prenatal endocrine environment is linked to the development of aggressive behavior and temperament. Temperament is biologically rooted, but it's adaptive to environmental demands. So remember that slow to warm, easy, and difficult is what we're talking about. Verbal and nonverbal aggression were higher in children whose mothers had low prenatal hormones as opposed to those who had medium or high hormones. Hormones, emotions, and parenting attitudes are related to activity level, reactivity, soothability, attentional focus, and high pleasure and fear. Okay, let's talk sex differences. So we're going to talk about cocaine again. Cocaine affects inattention and inhibition more in males than females. And size of brain, males tend to have larger brains. So they talk about these sex differences, and they don't really talk a whole lot about how that relates to disruptive behavior disorders. They basically say that they're not sure how these sex differences relate to disruptive behavior disorders or the development of them. They do know that the sex differences that are present at birth increase through childhood and adolescence, but the extent to which these structural differences contribute to regulation of behavior and emotions is unknown. So they do talk about it, but they don't give a whole lot of information. You'll kind of find that throughout this textbook where they mention something and they just kind of mention it and they don't really give any information about it. So that's it for this chapter. There are some additional readings for your assignment. Your assignment video will be posted as well. If you have any questions, feel free to email me, and I'll see you next week.